All right, our next section is going to be on transport. We're going to be talking about the different mechanisms of transport that are available to us in life. So basically what transport is the movement of materials from one area to another area. Now, in small organisms like unicellular ones, transport only happens across the cell membrane. But in larger organisms like humans, we also have another way of moving materials, and that would be our internal system, which is our circulatory system or our cardiovascular system. We're going to be focused on the transport that happens across the cell membrane in this section of our unit. The first type of transport that we're going to look at is passive transport. And passive transport is basically any transport across the membrane that doesn't require energy. In it, we are moving from areas of high concentration to areas of low concentration. And we have three types of passive transport that we are going to look at: diffusion, osmosis, and facilitated diffusion. So with diffusion, because it is passive, remember there's no energy, and cellular energy is in the form of ATP. So no ATP is required in order for this to happen. What happens is things will move from an area of high concentration to an area of low concentration, kind of like rolling a ball down a hill. It doesn't take any energy to do that. But once things have kind of equaled out, that's what we call equilibrium, the molecules don't just stop. They continue to move in both directions across the membrane, but it's kind of a trade-off. So one moves out, one moves in, one moves out, one moves in. So it always keeps the same concentration on either side. The factors that affect how well something will diffuse, there are certain things and certain characteristics of molecules that make it very easy for it to come across the membrane or hard to come across the membrane. So one of them is lipid solubility. So if something will dissolve in a fat, it is more likely to be able to diffuse across the membrane than something that doesn't. So this does play a role. The bigger thing that plays a role is the size of the substance. So things that are small are able to diffuse, whereas things that are large cannot. So if you think to your diffusion lab that you did with your model cell, the glucose was small, so it was able to diffuse out of the little model cell, whereas the starch was large and it could not. The second type of transport we want to look at is osmosis. Osmosis is the diffusion of water. And the cell membrane freely allows osmosis to occur. Sorry there, I had Mr. Phillips walk in on me here. All right, so osmosis is the diffusion of water. Now, we can look at how osmosis affects cells. So if I place a cell in a solution that we call isotonic, isotonic means that it is the same strength. So the water concentration on the inside and the outside are the same. Basically, if you have something that sits in something that is isotonic, you would see no change in the cell. If we put it in a situation where it is hypotonic, so the cell has less water than the stuff on the outside, the cell will swell as the water diffuses in. The way I remember it is hypotonic has an O, so it the cell kind of blows up like a big letter O. The other one is hypertonic, which means that there's less water there's more water on the inside and less water on the outside. So water will leave the cell. This is still a high to low scenario. So high on the inside, low on the outside, and the cell will shrivel as the water diffuses out. We can see how this works really well in the diagram that's in your notes there. This is actually a red blood cell, and the shape of the red blood cell is very important to its functioning. So when it is isotonic, we have things moving in and out. The purple dots are like a solute of some sort, and the blue are the water. So everything is moving out, and it is moving equally. And you can see down here with the electron microscope, those red blood cells are happy and functioning. In the hypotonic situation, we've got the arrows all pointing in. It's all water on the outside. So water rushes in and it blows it up like a balloon. This red blood cell can't function this way. In the other scenario, we have a lot of solute and very little water on the outside, so water actually rushes out of the cell and it shrivels it up. And again, that red blood cell will not function very well. So we are going to look at next the diffusion and osmosis worksheet that is in your handout packet. Diffusion is the movement of particles from an area of high concentration to an area of low concentration. Osmosis is the diffusion of water through a selectively permeable membrane. Look at the beakers on the left. In the beakers on the right, draw any changes in the water level or the number of solute particles on each side of the membrane that occur as a result of the described process. So if we look at the first one, we've got equal water levels on both sides, and we're going to have diffusion of the solute particles taking place on this one. So we also have to kind of look at what is happening. So because I'm diffusing the solute, I'm actually not going to move my water level. So my water level actually stays the same on both sides. But what happens is I end up with an equal number of solute particles on either side. So I drew 8 on each side. That's roughly about the same amount. So that's what would happen. The solute particle, some of them will move from the left side over to the right side, and they will equal out on both sides. In the second diagram, we have something different taking place. We have osmosis. And notice that the water movement here is going to go the other direction. So on the left side of the beaker, my water level will be high, and on the right side of the beaker, my water level will be lower. Now I'm still going to have my solute particles on both sides, but what moving the water does is it effectively equalizes the concentration. All right, and the question with this one. Look at the top left beaker. What would happen if the membrane did not allow water or solute particles to pass through? So if it's not letting anything through, nothing is going to change at that point. We're also going to take a look here at the next worksheet. Now what we have to keep in mind here is that those open, these that are here, oops, sorry, go back here, that the open ones, these are salt, okay, and then the black ones are water. So in the first one, I've got all the water at the bottom and all the salt at the top, and over time they spread out. This is diffusion. In the next one, actually it's kind of a little of both because it's diffusion of water, so it is osmosis as well. Sorry. Usually they label things the other way, where the water is the open and the salt would be the dark, but that's not what happened in this case, so that's my bad. All right, if we take a look at the next one, we have a semi-permeable membrane and all the salt is on one side and then it spreads out so that the salt is equal on both sides. That is diffusion. And then in the last one, we have the semi-permeable membrane with the water partially on one side and then as well as on the other, and it evens out. So this is osmosis. So those are the two main types of passive transport, but we also have one other type left, and that is facilitated diffusion. Now, facilitated diffusion sometimes gets confused with active transport. It is not the same, and we will kind of contrast that here in a little bit when we talk about active transport. But what happens in facilitated diffusion is we use protein channels that help move the molecules across the membrane. This is usually because either they're a little too large or because they're not lipid soluble, they can't get across on their own. The molecule binds to a receptor site on that protein channel, and then the protein changes shape. And this is one of the reasons why people tend to think that this is a form of active transport, because when you think of changing shape, you think of using energy, but that does not happen here. This is totally without energy, no ATP. The molecule is then released either to the inside or to the outside of the cell. And again, because this is passive, no energy is used. So we have a diagram here in your notes showing this. We're going, and this works very specifically with glucose. You use the protein channel to go through the cell membrane from the area of high concentration down to the area of low concentration. And this one actually shows how the protein carrier molecule can change shape. So the glucose molecule comes in, attaches to that receptor site. You can see the protein changes shape, so it closes off the other end, opens up the other, and then releases the glucose on the inside. So those are the three forms of passive transport that we have. We also have active transport. And in active transport, we're going the opposite direction. We're kind of climbing the hill. We're going from an area of low concentration to an area of high concentration. This type requires energy in the form of ATP. So some examples include things like endocytosis, exocytosis, the sodium-potassium pump that we use in things like muscle and nerve cells, and also water pumps. We're going to look here first at endocytosis. In endocytosis, materials on the outside are packaged to import into the cell. The package itself is called a vesicle, and basically what a vesicle is, it's a small sac that forms at the cell membrane and it gets brought into the cell. So this diagram here is a little bit more complicated, but what it shows is how endocytosis takes place. You can see here we start to get this little divot at number 1 in the cell membrane, and that forms a little pouch. And you can see here, all we need to do is pinch off that top, and that's exactly what happens as we get this vesicle that comes in. It comes in, we pull out what's on the inside, which is what we want, and then we can actually put the receptors right back up onto the cell membrane and repeat the process all over again. Pinocytosis is a form of endocytosis, and it is called cell drinking. That's literally what it means. There's no receptors that are involved here, so it's a little bit more simple. Basically, the vesicles form, but instead of having like those little dots, there's just a fluid there. This is very much less selective than other types of endocytosis. One type of endocytosis that you may hear a lot about, especially when talking about like white blood cells, is called phagocytosis. This means cell eating. I kind of call it the Pac-Man effect. These kind of cells kind of go and gobble up stuff. Basically what they do is they produce vesicles that contain solid objects, and those solid objects can actually be as large as the cell itself. And again, we use this, I said white blood cells, we use this as an immune response to foreign objects. So we take a look here. This is the process of phagocytosis. And if you look here, you can kind of see how it looks like a mouth that is eating here and how it's pinched it off. And that's the process of phagocytosis. Once it brings it in, it has created a vesicle, but let's say this is a white blood cell. It actually can destroy whatever foreign object is there and then take out the undissolved residue and eject it from the cell. Exocytosis is simply the reverse of endocytosis. We create the vesicle inside of the cell, and we actually kind of spill its guts out into the outside. Some things that we use this for are things like hormones, mucus, and even waste products. So let's make sure that we can contrast facilitated diffusion and active transport. So we are going to look at the next handout in your packet. Facilitated diffusion occurs when a substance diffuses across the cell membrane through a protein channel. Active transport occurs when the cell uses energy to carry a substance across the cell membrane. So we're going to look at the diagram. So we have this diagram and we have the one on the right. And it says label each as either facilitated diffusion or active transport. So this one, the first one, you've got glucose molecules coming in and going right on through, but what we are lacking is energy. There's no ATP expended here. So this one is facilitated diffusion. Whereas if we look at the next one here, notice that we have the energy going on here. That's ATP. That tells us immediately that that is active transport. Even though the process looks very much the same, because we have that energy in there, we know that it has to be active transport. So which process can move molecules from a lower concentration to a higher concentration? So we're going from low to high, we're going up the hill. That is active transport. Which process does not require energy? No energy, if you look up top, that is facilitated diffusion.